to introduce our first QSBC speaker of the year. It should have been the second, but last week's got canceled. But now we're fortunate to have Bishal here with us. He actually was part of our lab for a while, so it's a good homecoming for him. So he came to us indirectly from Nepal and Idaho, the College of Idaho, where he got his degrees in chemistry, engineering, and mathematics and physics.
Chemistry.
All of those.
Well, chemistry, math, physics was combined degrees.
Yeah. That was like a whole lot of degrees. Then he came to Vanderbilt and was in the CPB program and joined Vito Caranta's lab and was super productive. He was authored 9 papers while for the work he did while he was here. 5 of those as first author. He did a brief postdoc here with us after he graduated, but it didn't last long, right? So a year, yeah. And then moved on to University of Virginia to work in Kevin James's lab. And I'm sure the work that he has done here and looking at the single cell biology and understanding the melanoma, the diversity of responses that melanoma has to treatment has done him well in the environment with Kevin James's lab. It was also cancer focused, but a lot of single cell biology. So I think Kevin welcomed him with open arms. So looking forward to hearing how things have been going.
All right, thanks, Darren, and thank you so much all for coming. So I am a postdoc in the Department of Biomedical Engineering at UVA, and I also work very closely with people from UVA Comprehensive Cancer Center. So I'll present my work, two projects where I'm working on understanding the molecular variability that give rise to divergent cancer phenotypes. All right. So how many of you have seen this Hollywood classic, Fantastic Voyage? So the plot for this starts with they try to convert themselves into this miniature version of themselves so that they could flow into the blood vessels to help the fellow scientists. When I think of this, I think the possibilities that you could explore if you could flow into each and every cell and characterize them and understand them at the single cell level. Fortunately, all of these has, in many years now, since 1966, has become a reality, right? With the technology that we have at our disposal, we now understand and characterize biology at many scales. Often at single cell level, information going up from cells to tissues to organisms, from microscopic level to macroscopic level. We have information within cell for molecules and genes and whatnot. So things that we have understand with this fantastic voyage that we are in single cell biology and other technologies that have come up is biological systems. They span several scales. And information flows up and down between these scales. That's interactions between molecules, genes, interactions among cells in a collective system that give rise to emergent phenotypes. We also know that at each scale, biology is highly heterogeneous. If you look at cells, tissues, organisms, and whatnot, you'll always find differences at the phenotypic trait that you look at. And that has been known for many years. So this frequency and looking at the homogeneous distribution is not a new thing. What is interesting at times is when interactions among molecules or interactions among cells in a collective system give rise to qualitative differences at the population level. And that's where it becomes interesting. It was in the similar cells, identical environment. Now, the variability that you could quantify or identify giving rise to these phenotypic differences. And that could be 2 traits or 3 traits or multiple traits, depending on how many phenotypes that we have. So the major question that drives my postdoc work is how does quantitative molecular variability give rise to qualitative phenotypic differences? And I'm going to come to this from 2 different angles. I'm going to talk about part 1, where we look at the cellular basis of heterogeneity, and kind of understand what is driving the single cell differences and how they collectively give rise to the emergent phenotypes. And I want to, in the part 2, come back from the other side and understand what are the biomolecular differences that give differences or different outcomes in patients. So part 1, we'll look at the cellular level, and part 2, we'll look at the tumor level. And briefly at the end, if we have time, we'll talk about what I see myself doing, eventually understanding the role of nucleocytoplasmic transport, which came part of the work from the part 1. All right. So as I said, in part 1, I'm interested in understanding the drivers and mechanisms of tumorigenesis, at least from a cellular point of view, differences that drive some pre-malignant lesions to malignant, and some pre-malignant lesions that restrain themselves to become to stay at that position. So what is different about some lesions that go on to exhibit this cancer phenotype that is deadly versus some pre-malignant lesions that go on to restrain themselves? So from a systems biology perspective, we have seen this hundreds of times. There is a lot of characterization identifying genetic circuits, either naturally or engineering them to quantify differences at the gene expression level, or identifying some circuits looking at the response to stimuli. And many of these phenotypes are characterized at the single cell level. And we know either the genetic gene expression differences or responses are heterogeneous, and they give rise to this divergent phenotypes. What was interesting is at the multi-cell level or multicellular phenotype has not really been explored much. This classic paper that was published from Joan Brugge's group in 2001, they tried to look at this from a receptor level in a breast epithelial system. So they engineered this HER2 ERB receptor. When they're dimerized with this synthetic ligand, it creates, under dimerizer condition, this multicellular phenotype that comes as this highly disorganized, irregular phenotype. And the same when it's not dimerized is not that is mostly roundish. It does not have irregular shape that the dimerized condition gives rise to. So what is driving these phenotypes at the molecular level has been elusive over many years, and this is what we tackled on. So this dimerized ERB receptor system has this domains that can be activated with the synthetic ligand when you activate them. And if you follow the in a 3D system, if you follow the 3D clones, then you see some of them stay roundish, although they grow in size, while some of the clones go on to exhibit this multicellular disorganized phenotypes. And when we quantify this phenotype, we see, and this phenotype resembles, so this phenotype resembles what is called a DCIS escape in HER2 system, where DCIS is one of those HER2-driven pre-malignant state that stays as pre-malignant and does not really progress. That would be representative of these clones that actually become roundish and do not actually become irregular. But when you look at this irregular stuff, and they could be one step away from the DCIS, and when you quantify them, what you see is at the phenotypic level, the percentage of these DCIS phenotype that we see is 1/3. So 1/3 of the 3D clones that we observed go on to exhibit this multicellular disorganized structure. And we see this at multiple time points, and we found that the phenotype is actually obvious or distinguishable at 48 hours post the dimerization of these receptors. So to understand what is driving this multicellular phenotype at the gene expression level, we profiled gene expression at 24 hours post dimerization, and we did randomly selected 3D spheroids, and we got the transcriptomic 10-cell transcriptomics done in plus-minus activation with AP. And we matched the gene expression changes to the frequency of the phenotype. And what we found was many of these genes had biological significance in nucleocytoplasmic transport. Some of the genes that we found were genes regulating the nucleocytoplasmic transport, so transport between cytoplasmic nucleus, mostly importance and exportance. And when we looked at one of the regulators called CSC1L or exportin, when we overexpressed that regulator, we found the frequency phenotype with the dimerizer significantly goes up, and the converse is true when you do loss of function studies. But then we also had other regulators of nucleocytoplasmic transport, so it became really intractable for us to manually go after each of these. So we decided to look at how is nucleocytoplasmic transport altering this ERB dimerized induced multicellular growth through a systems biology approach. So we looked at, this is a work that Ian published while at UVA, back-to-back papers in, I think, molecular systems bio and JCB. So we decided to, so we basically reconstructed this systems model of nucleocytoplasmic transport. And many of these were in the context of HeLa cells. So we had to equilibrate the context in our system. And we're able to first look at the differences of all these regulators that we found to be important in driving these phenotypes. And for any nuclear cytoplasmic transport to be relevant, you expect the nuclear import with importance to be driving molecules into the nucleus, and there is nuclear export that is driving molecules out of the nucleus, and there is RAM gradient that directs this bidirectional flow. When we calibrated this model to our cells, and we looked at, under these initial conditions, what are differences in the classical nuclear localizing cargos, we found there is this divergent accumulation of nucleocytoplasmic ratio. So you see many of these under the same conditions have a higher abundance of these classical NLS cargos, and some of them have like really low abundance of these cargos. So what is different about this, and what are the associations that we can identify that drive these spheroids differently from the other spheroids? So when we look at the association of this nucleocytoplasmic ratio of classical NLS bearing cargos, we found that the abundance of CSU1L or the exportin, XPO2, is anti-correlated, and levels of importin alpha is positively correlated with the ratio of these classical NLS bearing cargos. But at this point, we knew that receptor activation was altering the nucleocytoplasmic transport with changes in the nucleocytoplasmic transport regulators and their abundances, and there is differences in how these exportin or importins were controlling this phenotype. We didn't know what were the cargos being localized or mislocalized in this process of receptor activation. So we went after that question, and we did biotin proximity leveling assay to identify what are the protein cargos that are bound when the receptors are activated. So we did this in two settings. So for the way this assay works is you fuse the protein of interest with biotin ligase. You then add biotin, which biotin lets the interacting proteins within the interacting radius, and you lyse cells, and you do mass spec. You identify many interacting proteins. Importantly for us, when we did this with biotin fused CSC1L, one of the regulators that we had functionally shown to be important in this context, and NOP37, other regulator involved in nucleocytoplasmic transport, we found many regulators of that were interacting. Most importantly, what came up was both EGFR and HER2 and a scaffold protein to be interacting partners to this. It suggested to us receptors themselves could be cargos. And we're not the first one to see this. Actually, there were multiple papers from this group in MD Anderson, which showed receptors could translocate to the nucleus, and HER2 receptors or ERB receptors, they actually have NLS-bearing regions in their sequences. So we cloned the NLS-bearing NLS-bearing sequences of EGFR and HER2, and we did we dimerized them, and we looked at the immunofluorescence. And what we found was within first 15 minutes of dimerization, there is significant increase in the abundances of dimerized receptors in the nucleus. So we're looking at the co-localized nuclear intensity, and it goes significantly up. One of the ways you could think of receptors getting in from the receptors to the nucleus is endocytosis. So things mostly mediated by clathrin. So this is a classic model where it gets into this clathrin-mediated endocytosis, cytotic vesicles, and get into the nucleus. So next, we questioned what happens if we block this process right here. Do we see changes or alterations in the phenotype that we see at this 3D growth level? So we knocked down the clathrin heavy chain with short hairpins, and we did that with 2 short hairpins. And in both of these contexts, what we saw was the knockdown of the clathrin heavy chain significantly increases the phenotype frequency that we saw. So it's basically suggesting somehow the receptors getting internalized are repressive in nature. So if you basically block it, now they lose that inhibition and the phenotype frequency goes up. And this was really interesting. So how do receptors first inhibit this phenotype? So the classic hypothesis, or the summary at this point basically was, when the receptors are mostly retained in the cytoplasmic region, they promote this outgrowth that we see, which resembles what we call an escape from DCIS state in HER2-driven cancers. And when they are nuclearly driven, they would inhibit this outgrowth. So we wanted to see what is happening with the receptors getting into the nucleus. So we performed chip sequencing, and we saw many significant genomic binding regions. Most interestingly, we're very intrigued by this observation that we saw was when AP or dimerized condition, one of the genes that significantly went up in the abundance was the microRNA called MIR205. And when we looked at the chip sequences, we found this was significantly enriched. So receptors were bound to the microRNA that regulate the expression of many genes, negatively regulates the expression of many genes. For us, it was interesting that this microRNA has binding sites for importance that would be responsible for bringing cargo into the nucleus. So it has a binding sites in 3' UTR region for KPNA1, also important alpha 1. And it was interesting because the hypothesis would be then that the dimerized context, there is activation or increase in the abundance of MIR205, which would then block the abundances of important alpha, so that the receptors are not really internalized as much. So we looked at the KPNA1, and we found that KPNA1 expression in my dimerized context is significantly reduced. And when we knocked down, so when we ectopically overexpress KPNA1 that lacks the binding site for this microRNA, we found the phenotype frequency significantly goes down. And this is the same setup that we have explored with dimerizer and DOX-inducible overexpression of KPNA1. In this setting, the KPNA1 lacks the microRNA binding site. So this basically suggested to us that translocated receptors bind to this microRNA, and then they downregulate importin, which then limits the import of receptors into the nucleus, and that somehow has a function in how it controls the frequency of this outgrowth that we've been looking at. So from a mathematical point of view, we were wondering, we also did other biochemical assays to identify the EGFR HER2 dimerized system required the binding of both importin alpha and importin beta. So in a classical NLS bearing cargos manner, they get translocated into the nucleus. What we didn't know was what are the binding affinities of these receptors as a complex with these importans. So with the mathematical model that I described earlier, we changed the binding affinity, and we looked at the ratio, the cytoplasmic to nuclear ratio of these receptors as a cargo, and how the relative abundance of importance and CSE1L affect that ratio. So when we did this in a very simple system, simple architecture that we have, we saw that with the CSE1L, that is the regulator that we had the most effect in when we did the functional perturbation. The abundance does not really have an effect in how it regulates the cytoplasmic to nuclear ratio of this receptor cis cargo. We see some effect with if you increase the importin alpha, there is the ratio going down. When we incorporated the information that we were able to get with the biochemical assays, with the chip sequencing and other assays, what we found was with this negative feedback, cross-inhibitory negative feedback, now that acts with receptors getting internalized, them binding with microRNA inhibiting the importance that brings the receptors in, and exportin blocking that import. So when you encode that mathematically, what you see is now the importin alpha does not really have much effect in the cytoplasmic ratio, cytoplasmic to nuclear ratio of the receptor cis cargos, whereas CSC1L, the phenotype becomes almost like a switch light. So if you increase CSC1L higher in abundance, you see there is this switch-like changes in the cytoplasmic to nuclear ratio, which corresponds to what we saw with the frequency of this binarized outcome in DCIS looking outgrowth. So this switch-like behavior resembles what we see with overexpression of CSC1L and pushing them towards increasing the phenotype that we saw. And we also looked at the single cell level when we quantified the abundances of CSC1L, and we plugged those initial conditions into mathematical model to examine how the distribution of the phenotype would be under the initial conditions of those CSC1L quantified. We saw that plus AP, there is significant variability in the dimerized setting in the abundance of CSC1L, and the frequency that we identified by simply drawing a line at the median level was about 1/3, which resembles to what we saw at the phenotypic level. So at this point, we are interested to see what is difference in the receptors between primates and non-primates, especially in the context of any cargo getting into the nucleus. You would expect them to have a recognized NLS. So if the hypothesis that we have, if the NLS is mostly present, it would mostly be nuclear. So by looking at the protein sequences, can we infer the likelihood of a protein to be in nuclear or cytoplasmic region? So when we use informatics approach to look at all the different family of HER2 receptors, mostly with EGFR and RB2, whether in rats, primates or non-primates, we found that the informatic score for HER2 in primates is significantly higher compared to non-primates. So this basically suggests that this simple single change in amino acid is altering the likelihood of this receptors being in the nucleus compared to non-primates. So we cloned these receptors with the changes at 678 codon site where arginine becomes glutamate, and we examined how the nucleocytoplasmic ratio of these two NLSs differ in these two contexts. So what we found was RB2, when the sequences NLS comes from rodents, it's mostly between the two compartments, so that mostly distributed. But if you look at RB2 when it comes from human, they're mostly localized in the cytoplasmic region. So because they have lower NLS, so it is as predicted by the informatics that we performed. So now we went on to isolate single cell derived clones with a dimer of RB1 and RB2, where RB1 was kept constant and RB2 either came from rodents or human. And we went on to do the 3D culture experiment that we did to examine how are the frequency of this phenotype different in these two dimerized contexts, with one coming from rodents or one coming from humans. In a set of four clones that we isolated and went on to characterize, what we found was consistently if the dimerization happens in rodent context versus human context, there is significant increase in the phenotype frequency that we saw with this multicellular outgrowth in human context, which has a lower NLS, which is almost as predicted by the mathematical model and the other biochemical assays that we saw, that if the receptors are getting less in nucleus, they're promoting the outgrowth. If they are more in nucleus, they're inhibiting the outgrowth frequency that we see. So we did this with multiple other clones, and the results were pretty consistent. So this seemed to suggest that primate HER2 has a weaker NLS, and it promotes the multicellular outgrowth that we see by being dimerized with EGFR receptors. Yes.
The NLSs, they look like, are those like right in the structural membrane region, just inside?
Yes.
Yeah. Is that the only one, or are there other nuclear localizations?
They are the major ones in these receptors that are driving those. Yeah.
Can I ask from the perspective of the cell?
Sure.
And I've experienced this myself with symptoms I made. So you might note this NLS, these are the things that are highly positively charged. Brain interaction.
Right.
So if you have a protein that has this, it's important to membrane interaction. Act as an
Right.
Can you explain to me, I get the endocytosis thing,
Right,
but a nuclear form, an endocytic vesicle
Right.
is gigantic.
right.
How do you suggest get it apart? I could envision some way in which that's cleavage.
Right.
Like that.
No, that's a really interesting question, right? So the first observation that was reported for these receptors getting into the nucleus, they actually suggested that this is a full length, which is really difficult to believe if you think about how big these are. We haven't done this experiment to look at what are the cleaved products that get into the nucleus, but that would be something that is worth exploring. These are gigantic and they need to be through the pore, nuclear pore complex into the nucleus.
I'm not. Line, and I've heard them ask several thoughts.
Yeah.
Obviously.
Yeah. They're so this was mostly driven with this one group from Emily Anderson in early 2000, and kind of went away. But there is new literature coming up that suggests that they could be in the nucleus and they could be driving gene expression by binding with many genomic regions. Yeah, we haven't really looked at the cleavage sites and cleaved products that get into the nucleus, but that's something that is really worth pursuing. Yeah.
The mouse, my understanding based on this is that it's the strongest of the NLS.
Yes. Yeah. Oh, awesome. Yeah. That'd be cool.
Have you confirmed that there are no effects on endocytosis itself, and that it's not a change in potential downstream effectors that we have access to in different locations?
Can you clarify that?
So internalization is pulling it away from the plasma membrane that gives it access to a different set of downstream signaling. That internalization itself, if that is affected, may be leading to some of these.
Right.
I'm just curious if you confirm that, how much you show that it is purely the nuclear localization versus other potential.
Well, in this context, we did both genetic perturbation to block that receptor internalization with clathrin heavy chain. We also use pharmacological inhibition, which is a dirty experiment. We see the similar observation. In terms of what are the downstream effects that we see, we haven't really done a thorough experiment to clarify those.
One thing.
Yeah. Yeah, but this is the dimerized. These are like dimerized. So we're looking at co-localized signal between the two receptors. Yeah.
Well, wait, so one problem is it's overexpressed.
Right.
Stopping,
Well, these are when they're dimerized, I think we're carefully chose these clones, the clones that we have that had almost physiological levels of dimerized receptors. Yeah. All right, so the part one summary is we showed that this interplay between exportins, importins, and RB receptors creates this cross-inhibitory feedback that leads to this divergent multicellular growth with this architecture now that exhibits, receptors getting activated, internalized, and activating microRNA that blocks the importins to inhibit the outgrowth. We took this further and we looked at the knockdown of CSC1L in an in vivo setting, and we looked at the tumor size and shape and how irregular they were. And what we found was compared to the control, CSC1L knockdown tumors were mostly circular. And we're looking at circularity. So if you have a high number, then they are roundish. Circular, if they have a low number, then they are mostly irregular. There was a recent paper that came in 2018, and this probably could get some of the discussion that we had they looked at the ERB2 receptors in gastric cancer, where there was a mutation in 678. And when they cloned these NLSs, they found that this mutant creates what almost looked like the multicellular growth that we have been describing. That is not the case in the control setting. So from, by looking at the NLS, NES, you could identify what fraction of what proportions of proteins get into the nucleus or get out of the nucleus, simply using informatics approach. All right. So if you have any questions, we can take a few questions here. I can take a few questions here, and then we'll move on to the second part of the talk, looking at biomolecular measurements that are associated with distinct outcomes.
Yeah, so I may have missed it, but how did you measure that the receptor is binding to an mRNA? Is that nuclear?
So we did the chip sequencing, and we found that when the receptors, so we did IP with the dimerized receptors, and when performed chip sequencing, they were significantly enriched in the genomic regions of microRNA. In this case, microRNA 205. Well, it's enriched in that region, yes. I think the physical interactions in what capacity, we don't really know. All we can say is based on the sequencing that we performed and we saw, compared to the control, IgG control, we see in the dimerized IP receptors IP setting, we see significant peaks in the microRNA region.
I don't have a good conceptualization how that. Are microRNAs thought that really only exist in the nucleus, or
I think so. Well, so the export import works with proteins, messenger RNA, any other molecules that are in the compartments, right? So, but they're mostly functional for many of these regulators that need to work on chromatins. They are functional when they're in the nucleus, but when they are not in the nucleus, then they're not as functional as they should be.
Okay, I guess all these questions are leading me to, are you sure beyond reading about that the mRNA interaction is occurring in that nucleus and not occurring cytoplasm?
Oh yeah, I see.
Cytoplasm there, unless maybe you're doing that.
Right. Well, we are not doing that, and that's a good question. But yeah, I was going to say, I mean, we do whole cell lysate, but this is chip sequencing, which mostly looks at the DNA that is extracted.
And it's chemical processing,
yeah.
So the proximity based. So
Well, not exactly biotin, but you use cross-linking to extract DNA and you IP them. Yeah. But there could be, I mean, some, yeah. I mean, so if some of them are cytosolic, I'm assuming they would not be found in the sequence, the material that we send for sequencing. So mostly the DNA that we send, the enrichment that we see in those DNA that we send, I think most has to be from interactions at the DNA. Oh yeah. So what we saw was in this dimerized setting, the microRNA 205 abundance goes up. And you're asking if you overexpress microRNA? Oh, yeah. Yeah, we haven't done that. That's a good point. So if you do that at a basal level and see what happens to the phenotype, that's a good point. Yeah. Yeah, that's a good point.
So you induce dimerization, goes through the nucleus, finds microRNA,
Among other things, yeah.
Is that, you think that's associated with the outgrowth or in addition to that?
So if it gets into the nucleus, it is inhibiting the outgrowth. So frequency of the phenotype goes down. So if it is retained in the cytosolic region, mostly in the receptors, then it is promoting the outgrowth. So
if
the,
you take these clumps of cells.
Yeah.
Or like the treating on the diamonds. You can look at the ones that are not outgrowth and are outgrowth, can you see the things in the tension on these receptors
so,
on their own without having to use the system?
so first of all, we are doing this in a 3D with a dimerized. So in a basal like conditions, when you don't add the synthetic ligand, they are just normal MCF10A with a engineered system for EGFR and HER2. So for us to basically examine how this activation changes the signaling, we have to activate them, right? And you're asking, what are the localization under normal condition? Is that what you're asking?
Right, yeah, there's a difference between the ones that don't have an outgrowth and ones that do.
But they only exhibit the outgrowth when you dimerize them. So basically, yeah. So you activate these, you put the synthetic ligand, the receptors get dimerized, and the frequency only is observed when you and that was something that we see about 48 hours of post dimerization. So the phenotype is not even obvious before that. And one of the other things that I didn't really discuss much was when you take those clones out, the multicellular clone that we see, and you dissociate them and you perform, reculture them and put them back in the culture again, you see the same phenotype that comes up. So there is that reversibility idea that is happening in this, and I'm excited to explore that in further detail. No, not in this context. In this context, we haven't looked at any of those. Right. Yeah. We haven't really looked at in greater detail of where the microRNAs are. Well, the target in this context that we looked at was KPNA1. That seemed to be a lot of things. Yeah, that was the one that kind of was in the same vicinity of the nucleocytoplasmic transport regulators that we know. One of the other things that I have been really excited to pursue is microRNA MIR205 has also the clathrin heavy chain as one of the targets, especially from the informatics that people have done. So that would be really cool to see if it has a connection with the endocytotic regulators being involved in this process of microRNA going up and somehow inhibits the abundances of CLTC, and somehow that brings the receptors. Oh, it's conserved. Yeah, it is absolutely conserved, and that's something that I think we looked at. I haven't looked at it. All right, these are all great questions. Okay, so for the second part, I want to talk about the other end of the spectrum that I mentioned earlier. I want to look at biomolecular features that are associated with distinct outcomes at the tumor level. So this is a part of the large program project grant that is awarded to UVA Comprehensive Cancer Center. I've been working in collaboration with them. The grant was awarded to Tom Loughran, and we're looking at what are the differences between tumors and what implications they have in the clinical setting. So the program project grant, this program project grant focuses on AML, acute myeloid leukemia, and AML has been a subject of extensive sequencing or any other omics that you could think of. And all of that has basically shown this is a highly heterogeneous disease. And many of these genomic classification in AML have been proposed to either stratify patients and devise new treatment strategies. But two problems lack. Majority of patients still lack those genomic features, so there is always some proportion of patients that come in the clinic who don't have those known mutations, and the therapies have not really improved in 30 years. The focus for the PO1 grant is on the role of sphingolipid metabolism in AML. The sphingolipid metabolism has this really simple, I guess the simple schematic in this that I'm displaying. The major hub is ceramide, which needs to be converted to all of these different sphingolipid species based on enzymatic activity. The work within the PO1 group and others have shown that sphingolipid regulate whole range of cellular processes, although they are the integral member of cell membrane. And depending on the species and depending on which species are abundant, they are either apoptotic or pro or anti-apoptotic. So it is really interesting to think of how this balance is achieved in cells and how dysregulation of this pathway contributes to diseases, including cancer. So with this PO1 project, there was a lot of data with AML patient samples, AML cell lines, and this was collected in 3 different cancer centers, UVA, Sloan Kettering, and Penn State. And like I said, the cell lines and patient samples were sequenced for this biomolecular quantification of sphingolipids. So we call them sphingolipidomics. So we looked at different abundances, different species of sphingolipids and their abundances. And the objective for us was to basically see when you look at the patient samples and cell lines and their sphingolipidomics, can we identify some consensus clusters? Do they have similar features of sphingolipids that are similar and different in different patients and cell lines? And with some of these samples that were profiled for sphingolipidomics, we also have RNA sequencing done. So we wanted to connect this and see if the metabolomically defined clusters have some distinct RNA signature. With a goal to build a classifier, then that would look at the RNA expression, mostly because it is so abundant in public data sets like TCGA or BTML, and ultimately to be able to predict the patient outcome, which would connect how sphingolipid profile would dictate how patient would respond to the convention therapy. So when we looked at normalized lipid profiles of patients and cell lines and compared that to the normal CD34 enriched samples, we found that the patients and cell lines were mostly intermixed, but they were separate from normal. And there is a lot of heterogeneity, as you expect, in these samples in both patients and cell lines. That also gave us a motivation to examine whether there is a pan-AML classification that we could achieve by looking at sphingolipidome profile. So from the consensus clusters that I performed, we identified 2 statistically robust clusters that varied in the abundances of sphingolipids, mostly the sphingolipid species called sphingomyelin and hexosylceramide. So there is cluster 1 that is abundant in sphingomyelin and deficient in hexosylceramide and vice versa for the cluster 2. And none of the other sphingolipid species in broad categories that I'm defining were different between these 2 clusters. So like I mentioned earlier, some of these samples that we had for lipidomics, we also had RNA sequencing done. So I wanted to examine how, if we cluster them just based on the RNA-seq global expression, how would they be clustering patients and cell lines? And this is the sphingolipidomic best clusters where patients and cell lines are very intermixed. So we have a good abundances of patients and cell lines in both of these clusters. But if you do the clustering based on solely on RNA-seq data, what you see is you identify clusters that is mostly patients or mostly cell lines. So we think that sphingolipidome, a biochemical measurement, integrates patient cases and cell lines much better than global gene expression. And this is mostly because when you look at the global RNA sequencing, you quantify many genes that may or may not have direct regulation of different biochemical processes in these samples. So with the identification of these 2 robust sphingolipidomic clusters, we looked at how these clusters are different clinically in response to the chemotherapy that patients receive. So when you look at the response versus failure in these 2 subtypes, what we found is the sphingomyelin high group has a higher frequency of failure compared to sphingomyelin low cluster. And when you look at the event-free survival or overall survival, you see that the sphingomyelin high group, which is this purple, has significantly worse patient outcome compared to sphingomyelin low group. So with the gene expression data that we had, now this is very curated, right? So it's supervised because now we know these 2 groups were different in the biochemical measurements. Now we went back in a supervised way to examine the differences in gene expression. And what we found was obviously many genes that were upregulated in one cluster versus the other. But interestingly, we found that genes upregulated in sphingomyelin high group were enriched for the signatures, transcriptional signatures of leukemic stem cells. So leukemic stem cells, they are the cells that could differentiate into many other many other cell types based on this differentiation trajectory. And there were a lot of single cell gene expression data from published papers that have all of these different groups functionally characterized, or gene expression at the gene expression level fully characterized. So I went back and looked at our two sphingolipidomic subtypes and quantified the abundances of these different cell types with tumor deconvolution to quantify what are the frequencies and abundances of these cells in these two groups that we have. What we found was in the sphingomyelin high group, the undifferentiated cell types are significantly higher compared to the sphingomyelin low group. In contrast, the myeloid differentiated cell types were opposite. So this basically suggests that this clinically poor group have higher abundance of higher abundance of leukemic stem cells that is associated with the failure to the current therapies that exist in the clinic. So a sphingomyelin high group have a significant enrichment of leukemic stem cells in all of these different categories that people have defined, and then the sphingomyelin low group have mostly differentiated cells, and that could be associated with the clinical differences that we see in the clinic. So to take this further, because many of the public data sets don't have the sphingolipidome profiles, but they have a lot of gene expression data. So how can we infer a lipid profile of a sample based on the genetic? With the data set that we had with both sphingolipidome profile as well as gene expression, I divided them into trend and trust data set and validated the model. And with the selected model that we found based on the accuracy, now we projected we use the model to basically predict what the sphingolipidome profile of a patient sample would be based on the public repositories that we had access to. So the classifier did really well, and we wanted to see if the predicted patient samples in these two lipid profiles have differences in the outcome. And consistent with what we saw in our internal cohort of 141 patients, both in Beat AML, which has about 180 patients that had not received any treatment before, and in TCGA, about 150 patients. Consistently, the sphingomyelin high group does significantly worse compared to the sphingomyelin low. This was really interesting when you think of the clinical categorization based on the genomic information that clinicians get, and also chromosomal abnormality. Patients mostly get categorized into three groups: favorable, adverse, or intermediate. Intermediate risk group being very difficult to treat because it's highly heterogeneous, and it is often called a basket category because they don't really know what is driving those. So anything that does not fall into those two, favorable and adverse, falls into this intermediate group. When we projected the intermediate group into our biochemical projection, what we found was for the patients that fall into intermediate risk group with sphingomyelin high profile, they do significantly worse than patients that have sphingomyelin low. This suggests that this intermediate risk group, which is often considered a very difficult group to treat, can be further stratified based on the biochemical measurements that you could do by looking at their lipid profiles. So the summary of this, we found that sphingolipid focused metabolic profiles by biochemical measurements identified two distinct AML subtypes, and high sphingomyelin subtype is a previously unrecognized high-risk group with poor clinical outcome. And we looked at whether sphingolipid is an independent predictor of outcomes. And it is. If you look at the hazard ratio, it is significantly high compared to the baseline normal comparison. And we found that we have a lot of experiment and high throughput analysis going on to look at the differences in drug sensitivity for the samples that fall into these two groups at the cell line level, as well as the patient samples where we have high throughput pharmacological screens. And we found that these subtypes, depending on which subtypes they belong to, they have different outcomes in the inhibitors that we have used, mostly the lipid altering drugs that inhibit acetylceramidase or this drug that blocks the accumulation of ceramides by inhibiting the conversion of dihydroceramide to ceramide. So there is interesting, if you look at the differences, right? So a sphingomyelin high group is insensitive to this drug, but sensitive to the other drug. So there is this different outcome that we see in these two groups that could be a potential avenue to examine for drug combination studies that we are currently undertaking in the Cancer Center. All right, so how much time do we have? All right, so I'm going to go briefly. So one of the things that I found was when I looked at the metabolic clusters, and I identified the genes that were significantly regulating those two clusters, and I built gene regulatory network for each of those clusters, and I looked at highly connected genes in those networks. What I found was for one sphingomyelin high group, which is clinically worse group, I found many of the highly connected genes were transcription factors, epigenetic enzymes, or kinases that have the potential to be mislocalized or distributed between the two compartments. And between the two subtypes, and when I looked at the important beta expression, there's significantly increase in the sphingomyelin high group compared to the sphingomyelin low group. And the regulators of nucleocytoplasmic transport are also very different in these two groups. And this is really interesting and particularly, I guess, timely, because there is a inhibitor that is in the clinic that blocks XPO1, and this had huge success in myeloma, was FDA approved, but there is a lot of new observation or new results coming up that suggest that despite early success, this faces an uncertain path to expansion, mostly because blocking exportin, then you block a lot of cargoes that could be localized in the cytoplasm. So people don't really know which cargoes have, effect when they are mislocalized or localized in these two compartments. So one of the avenues that I see combining the earlier part of the project that I discussed and with this is a patient-specific nucleocytoplasmic transport models to look at the cargoes and signatures that could be different between these two subtypes, and in how that information could be used to stratify patients into resistant versus the sensitive groups. And this could be done by examining the public, by looking at public repositories for gene expression that could be fed into the mathematical model. And a repository like BTML also have the patient outcomes reported for the exportin inhibitors. So that could be an association that we could think of building. So there is the possibility of building the cell line or patient-specific models and using that information to stratify drug sensitive, insensitive from both, identifying the molecular features that are associated with drug resistance, and also what signatures are driving these therapeutic differences that is observed in the clinic. Yeah, this is basically like what I envision myself doing in terms of my future work is investigating how the dysregulated transport give rise to cancer vulnerabilities and emergent cancer phenotypes. And I'm interested to pursue this in two areas. One would be looking at the tumorigenesis, other would be looking at the therapeutic angle by building a mechanistic framework for transport inhibition. So with that, I would like to acknowledge people in Jen's lab, mostly Lishan, who was first co-author in the manuscript that we published, Anthony, who is now pictured here, and people from UVA Cancer Center, mostly Tom Loughran, Dave Fife, Todd Fox, and many collaborators that I have had pleasure of working with in the past. With that, I'll take any questions that we have.
How do you know that these nuclear transport inhibitors
So this drug that was FDA approved blocks the system residue in the NES binding site of XPO1.
Well, I mean, but targets.
Oh, which targets are blocked? Is that like about 500 different cargoes? Yeah, so that's the reason that these drugs actually have not really gained a lot of traction, because earlier on people are excited. The company that develops this, I guess it was fast-tracked by FDA to approval, but people don't really know what is happening in terms of this homeostatic homeostasis being, disrupted when the exportin is blocked.
Classic drug, like in any capacity.
I think that's why there's a avenue for a systems biologist, I guess, to come into this and to actually look at from a systems approach, right? What is happening, what types of cargoes, broad categories of cargoes, or focus them and identify which cargoes, when mislocalized or localized in one or other compartments, have a therapeutic window. But I mean, it's obvious that this drug is clearly not working, and people don't really know why. And the reason is there is existing heterogeneity in the nucleocytoplasmic transport state of different patients that people don't really know. So by using these transport models or extracting those signatures, you could basically say, I expect based on these predictions that these patients will not respond to this therapy. So there is a real opportunity here to use this systems approach in not only understanding what is being affected in terms of homeostasis, but also stratifying patients into categories of response, no response.
So it doesn't work in it works.
So AML in the early preclinical studies was really good, and when they took it to the advanced, it does, yeah.
There seems to be some sort of regarding this specificity that you envision that specificity arises at the expression level is important.
Yeah.
Important,
Yeah, right.
One important is really expressed in the so that's one layer.
Right.
This is a different level of strength of the animal.
Yeah. Right.
The really strong LS
Right.
versus a weak MS, right?
Right. Yeah.
Both of those are.
Absolutely. Yeah, absolutely. And you and I talked about this in the morning. I think there is a real chance that you could envision looking at the specificity of this exportins, the 7 exportins, right? Not only XPO1, we have XPO1, XPO2, XPO3. We found XPO2 to be relevant in breast cancer. I mean, all of these and the specificity of the cargoes that are being regulated, NLS strength that is different in different cargoes, which cargoes have potential to have this effect. All of these are worth exploring. Yes. So depending on the type of sphingolipid species they get converted to from ceramide being the central hub, if they get converted into the pro-apoptotic versus anti-apoptotic, there is a linkage with a mitochondrial mediated cell death. So some of these ceramide downstream of ceramide, they have protective effect, or so others have cell death effect. We have done some studies with the collaborators at UVA looking at how mitochondrial function is different between these two subtypes. So people have looked at OXPHOS capacity, glycolytic capacity, and connecting and identifying the respiratory potential for these two groups, and we have seen some differences. Yeah, there may be because the ceramides as a second messenger could be involved in a lot of things. So there could be other pathways that we haven't really looked at yet.
All right, thank you.
Thanks so much. How do I end this? Oh, so